Lisa holds a BSc from the University of California at Davis and started her career at the Immune Response Corporation studying biological markers of autoimmune disease. She then worked at Amylin, where she directed the amino acid analysis core facility and performed method development for peptide drug candidates. She then moved on as analytical project manager to ISIS Pharmaceuticals, where she worked on the chemistry, manufacturing, and controls of antisense oligonucleotide drugs. Currently, she has a role in Catalent Science and Technology team in San Diego, working with pharmaceutical companies to identify the right development pathway for the early development of drug candidates. And she's going to give a talk entitled Transitioning Peptides from Discovery to the Clinic. Lisa, thanks very much indeed.
Thank you. All right, thank you very much. So I'm here in two roles today. My first role is part of the advisory board for the Boulder's Peptide Symposium. This is my third year, and I absolutely love this symposium. I think it's great. We learn a lot. There's a lot of diverse speakers. There's a lot of diverse topics that we're talking about, and so I thank you for that. And part of that, I will be helping to monitor the workshop that we're going to do later. So here's my plug for everybody to come back from lunch and join us at that. That'll be a lot of fun. But my second role is here as part of CATALENT, right? And so some of you may be wondering, what is CATALENT doing sponsoring a peptide symposium? Aren't they the soft gel guys, or maybe they're the guys that bought Micron not too long ago? I will say they're peptides because they purchased my company, Pharmatech, about a year ago, and that is a company that focused on early phase development of small molecules and peptides. And then why I am here is because, as you heard, about 24 years ago I worked at Amylin, and that was a really interesting experience. It's kind of where I fell in love with peptides, if you can say falling in love with peptides. But also that experience with Amylin, I think, is going to translate into why we're having the workshop a little bit later about how maybe you can do things a little bit more virtual versus having 200 people to develop one molecule. So today I'm talking about transitioning peptides from discovery to the clinic. I think my audience today is really those peptide medicinal chemists, the drug discovery people, but also maybe some of you who might be doing due diligence on a peptide asset and might be trying to look at, is that asset really something that can become a drug? All right. Again, always like to set the stage. You know, we're in this area because we know peptides can be drugs. Did a little quick search, and we have over 300 companies, different companies evaluating peptides in preclinical and phase 1. And within those companies, more than 500 different programs with peptides. So certainly there's a lot of chances to do good, and there's a lot of chances to screw up. So let's try to do it right. Let's try to get drugs into patients. So I always like to talk about this because I do talk to a lot of early phase development folks and a lot of medicinal chemists, and they are so excited about their molecule. And I say, okay, we got a molecule. We need to make it a drug. So the FDA does have a definition, a legal definition of a drug. So we can read that here. I suggest that all of us are in this drug development world because we all believe our molecules are going to be drugs. So we're not doubting them. So let's start thinking really early in development that they are going to be a drug. They are going to be successful. They're going to get into patients. They're going to go commercial. So today I thought about how to break this down, and some of the things the way I thought I would break it down today is to look at some of the issues I've seen as far as peptides as molecules, and maybe talk to you about how we might be able to fix them a bit in discovery so that we have a drug. And I thought a little bit more about it last night, and you know, you discovery chemists are amazing people. You're doing all sorts of cool things. And so I thought about my role today as kind of like mom who's calling you in from the outside playing. It's getting dark, right? But it's time for dinner. Got to get down. We got to get fueled up. We got to make this long term. So I'm not the buzzkill. I'm not the bad guy. but there are some regulatory considerations that we need to think about as we're taking a molecule into be a drug. All right, so I thought it was easy because, you know, amylin. Let's talk about amylin. So, you know, you all as discovery chemists, you're looking at, you know, finding a peptide that's involved in a disease state. So amylin's a great example. You know, they determine, hey, here's a 37 amino acid, pancreatic islet peptide, so co-secreted with insulin. It's deficient in type 1 patients. Identify the structure, and bam, you got a company, right? You got amylin, same year. But they realized, as we just talked about in the last talk, that amylin itself is not a drug. You know, if you try to take amylin as a drug, it's a no-go. So smart people at Amylin said, hey, we got to make some changes. And so for them, it was actually quite brilliant and worked great. They were able to replace some amino acid residues with 3 prolines. You got triproamylin, you got pramlintide, you got Similan. So great little change, made a huge difference between a molecule and a peptide. So again, when you're thinking about your sequence and you're thinking about your sequence being a drug, there's other considerations you can make as well. And you know, we talked about the last talk. I don't want to go into it in depth, but you know, there's certain amino acids that you can have as part of your sequence that can make it a little bit more difficult from a chemical stability standpoint. We're thinking about CMC. We're thinking about shelf life. We want to get you that 2-year shelf life. So just think about the considerations of what residues are in your molecule. Do they need to be there if they're the more troubling residues? And if not, you know, maybe your potency drops a little bit, maybe your binding affinity drops a little bit. But if we can get a longer shelf life for your drug as the drug product, even if you have to make a bit more drug substance, it's all going to cancel out, and you can have 2 years, 3 years, 4 years shelf life of your drug. You're just not making drug product as often. Binding affinity, the molecule, you know, so important. Again, we touched on this. I always laugh because I know I'm talking to a peptide chemist because they talk to me in nanomolar and micromolar and picomolar. And as a formulator, I'm always trying to do the math and convert that into mgs per mil because that's what I care about. But it's great. At least I know where we all stand. So you guys know you need to get a drug. You want it to be potent. You want it to be hitting that target at low concentrations. But again, that can be an issue. Aggregation is a huge issue. It's a huge issue to the agency, right? They are very aware of this as a potential for adverse events in patients. They do not want to see an aggregation potential with your molecule. And one just real quick example I put up here was a project where they had taken the larger protein and they broken it down until they really got to the beta sheet region. And this region was great for binding their target, but it was also great to stack and gel. And it was very concentration dependent. And so you can do some things with formulation development, but really what you want to do is you want to go back to your sequence. That's really where you're going to make the fundamental difference about that gelling and stacking. And again, we can do some other things. We can do some fancy stuff with cyclodextrins. These are great little molecules where they have a hydrophobic center and you can shield them with their hydrophilic exterior, hydrophobic interior, and maybe shield some of that. But you know, really it's a problem. And again, I suggest, you know, let's look at the data. If you have 2 molecules, one gels and stacks, one does not, and they're similar in their binding affinities, it might be worthwhile to go with the one that gives you a little bit higher binding affinity, but you know, it has better drug properties. Binding affinity adsorption, very practical. Analytical challenges, we all know. We've all worked with them. It's sticking to your glassware, it's sticking to your vials, it's sticking to your filters. I've seen it stick to Carl Fisher apparatus, so we're trying to do accurate water content measurements, and the drug's sticking to the Carl Fisher electrode. We can't get accurate data for you. But again, more importantly, are some of the manufacturing clinical dosing challenges. We need to get the right amount of drug into the vial. We need to get the right amount of drug out of the vial into a syringe, into a pump, and then into the patient. And we need to make sure when it goes into the patient, it's not going to aggregate. You're not going to have adverse events. TFA salts, as far as the molecule goes, wow, very easy to make. Every peptide synthesis house is probably going to give you a TFA salt form. It's easy, isolate it right off the HPLC. And again, kind of what Mark said. But why not the TFA salt? You know, besides potential for toxicity, besides issues with the in vitro assays, why do we not want to do it? You might be able to get away with some early PK work with the TFA salt. When we dissolve that up in a vehicle, it's going to be a very low pH. So we're going to have to work hard to get that back up so it'll be physiologically compatible. So there's additional work there. I suggest too, though, that TFA is a big molecule. It's bigger than acetate, twice the size about. And you're paying for it if you're manufacturing it, if you're paying for bulk peptide. So smaller counter ion, less expensive. As far as the GLP in clinical work, we do need to be in the final salt form. We want to make sure that we're using the same form in our GLP as we are in our clinical work. And again, when you're picking a salt form, you do want to consider things like manufacturability. And then, as mentioned, the counter ion can influence the degradation of your API, potentially some of the interactions in the drug product, be it solution, be it a lyophilized product. So all those things are in consideration for counter ions. This gives me a chance to do a side discussion with a passion project of mine, which is how we're all communicating about our drug as far as what is our drug. And I do reference down here the FDA guidance on salts naming. We need to be talking about the active moiety that you are working with. And we need to be careful that we're not talking about bulk, we're not talking about net, and we're not talking about it as a salt form. We're really looking at what is in that powder that you are providing us, what is the active acting portion of it? Because I have had situations where people in early toxicology studies are all dosing based on bulk powder. And you guys realize in early phase development that bulk powder may only contain 80% active peptide. And that 20% can be very meaningful when we're looking and trying to figure out how to be setting the clinical dose, et cetera. So if we all start talking about active peptide at the same time, we're all going to be talking the same language. So there's two great ways to do it. One is taking your bulk peptide, multiplying by the peptide content, multiplying by the HPLC purity. I sometimes kind of like this version with that water content being a variable that we can assess at the time of use. And I'm going to talk a little bit more about hygroscopicity. But I just, again, suggest we all talk about active peptide. Phosphate buffered saline. It's great. It's on your shelf. You're using it for some of your in vitro assays. It's non-toxic, isotonic. Why should we not be taking that into the clinic? A couple things. So drug solubility. Sometimes sodium chloride, again, we saw it in the last talk, sodium chloride can have a salting out effect, and you can actually reduce the solubility of your peptide. Manufacturability. Phosphate is poor in lyophilized products. It can have the tendency to crystallize out. You can get these pockets of low pH, and you can actually see variable degradation of your drug product, and it could be vial to vial. So you do not like that amount of variability. NaCl itself is a poor tonicity adjuster for lyophilization. It's going to lower the collapse temperature, potentially makes you have a very long, low temperature cycle. Not great for when you're paying per day for your lyophilization cycle. As far as container closure compatibility, I don't know how many of you were part of the 2010 great EPO recall, but that was very hard on the rest of us that needed to get glass, right? So EPO was recalled due to delamination of glass. And so all the rest of us, especially in clinical development, were very low on the totem pole for getting glass vials, and so we don't want to run into that. In addition, phosphate buffers themselves have a unique interaction with glass that can happen between the phosphate and the glass, and you can see particulates. Slightly different mechanism, but you can see particulates, and I have seen them in several products that have been brought to us. Mitigation, there is a non-glass vial option. Here's my CATALENT plug. We do have things like blow-fill seal and Adsorption. These are polymer-based container closures. Usually for phase 1, though, you're going into glass, and you do want to have that consideration. But of course, one of the easiest things to do is just look at different buffer species. Great thing. Histidine is a great buffer for peptides. On the research side, you're very excited when you get a highly soluble compound. It makes your assays a lot easier. You don't have to struggle. There's no dose-limiting precipitation. As far as the drug goes, though, if that high water solubility translates into hygroscopicity, where your drug is taking on water, it causes us a lot of problems. Just routine handling. If your water content is changing as you're using it, all your quantitative analytical results could be in question unless you are either controlling the environment you're weighing on it, or if you're doing water content at the time of use. You may need to implement a reference standard a lot earlier, and that's a working reference standard. So it might be something where you take vials and you lyophilize 1 milligram in 100 different vials, and then that's your working reference standard. Because if you don't, it could be the water content could be changing on your solid reference standard at any point. And then, of course, there's just accuracy during compounding. When you're weighing the amount of material that's going to go into your compounding vessel, we need to know how much water is in there. Otherwise, you potentially have to do an in-process test to make sure that you made the right amount. If the in-process test comes back off, you're going to have to add more material. So just a real quick example of that. I just want people to think about a hygroscopic peptide in terms of their COA water content. That water content on your C of A is the water content that moment in time for that aliquot of peptide that you were testing. When you start shipping that peptide off to other labs who are opening it up in the atmosphere, maybe in a hood, we don't know, it's going to be different. So if you're trying to take the water content for any calculation off of that, it's probably not going to be right. And also keep in mind that the peptide content that's on your C of A is associated to that same water content value. So again, as the water content changes, the peptide content value that you rely on so heavily is changing as well. And then again, just to touch back on the reference standards, if we did have to use the powder as a reference standard for any quantitative work we're doing in a more QC environment, we're probably going to have to do a concomitant water content measurement, and that could be up to 50 additional milligrams of reference standard that we're using every time we do an assay. That's expensive, right? So we don't want to do that, and that's why sometimes it's easier just to take the hit, go to the vialed working standard, just grab one out of the freezer, use it, and go. All right, chemical modifications, and that's one thing about this conference. It's so great to hear about all the great stuff that's going on to improve half-life, cell penetration. You're targeting. You're doing some really cool chemistry. From the drug side of things, there are challenges, and this is just a little limited list. But of course, as people add more hydrophobic groups to these molecules, we're running into hydrophobicity issues. We're having to formulate them more like small molecules than peptides, and that leads to some other issues. Adding co-solvents is fine, but when you start adding co-solvents and you're manufacturing in a vial, you have to start worrying about things about extractable leachables, those types of issues. So we try to keep co-solvents out unless we absolutely have to. From an analytical perspective, simple things like polydisperse profile of a pegylated peptide, how do we control for that? And then this is a significant one, is as you are sticking larger hydrophobic moieties onto a peptide, that will start modulating how that peptide is interacting with our reverse phase HPLC column, and that interaction is going to swamp the ability to see some of the more subtle changes in the peptide itself. Simple things like not simple, but things like deamidation. So we might have to look at disassociating that hydrophobic species from the peptide to be able to accurately characterize what is happening to just the peptide itself. A little plug here, though. The cyclized peptides I've worked on, those are actually a little bit easier. They are a bit more like small molecules, so from an analytical perspective, they are not quite as difficult as putting a large hydrophobic species on there. Back to our old friend TFA. TFA is usually the go-to modifier for HPLC method development. And it's great. It's easy to prepare. You get great peak shape. It's a mild ion pairing agent, so it works great. Some people say it's mass spec compatible. I'll let you decide if you want to put it on your mass spec, clean it up a little bit. But anyway, so it's a great place to start. The thing I want to remind everybody is that TFA is not UV transparent at our normal wavelengths that we're monitoring, right? So when we're looking at peptides, we're usually at 210, 215, 220. It's not UV transparent. So what does that mean? So again, we're thinking about regulatory environment, quality control. So this is my tale of two modifiers. So this is the exact same molecule. The upper one, that is run in a TFA mobile phase, and the second one is a perchlorate mobile phase. So just looking between the two, the perchlorate, which is transparent, we detect 3 times more peaks in that chromatogram. And you may say, I don't want to see all those peaks. And I suggest you do want to see all those peaks because you want to know what's popping up. You want to know before it all of a sudden hits a limit or hits some sort of qualification threshold, right? So I want to see the gradual increase so I can actually plot that in a linear fashion, maybe do a little Arrhenius kinetics, and understand what my shelf life is going to be. So if I don't see them, if I'm blind to it, I can't do those studies. And so if you look here, we have 10 times the sensitivity. I suggest the peak shape is a little bit better too with the perchlorate. So yes, you can go into phase 1 with a TFA method. It's not going to kill you. I just suggest that you might want to start looking at some alternate modifiers to have a bit more robust analytical methods that are going to be easier to validate, that are going to be a bit more regulatory compliant as you move along. And I know, sometimes people say, but the ICH3A is not, says no peptides. And what I would suggest to you, though, is that the regulators don't have a guidance on peptides to go to. All they have are these ICH guidances. So even though the guidances say, peptides are excluded, they still need something to root to. And because peptides are treated a bit more like small molecules, certainly more so than a biologic, they're going to look at the guidance. So here, just for everybody remembers, is that we're usually looking at reporting thresholds of 0.05%. That's from your analytical method. And so we want to have sensitivity down to 0.05% if we can. And again, this is another example of a chromatogram. I mean, I look at this. This is TFA at 220. Here, the limit of quantitation is 0.1%. I can see 3 peaks here. There may be something going on over here. If I ran it enough times, I might be able to get an idea about whether they're real or not. But certainly, they're not more than a signal-to-noise of 3 or a signal-to-noise of 10 to be at the limit of quantitation. So, it's a little quick, but hopefully I was able to talk to you about some of the things that you might be thinking about in discovery, putting it on your radar screen as to what we might be needing as we're developing your product, your API into a drug product. Let's start the conversation early, earlier the better, and hopefully we can kind of break down those barriers. All right, that's my talk. Thank you.